Good morning, student. Today I am going to start chapter number 8, the cell. After a long gap I am giving the video. Several students of class 8, this video is dedicated to class 8, but higher class also will get some basic concept, and today I will explain about this chapter number 8, cell. The cell is the unit of life. Today I will explain all the things and all the internal structure of the cell. After seeing, you see several times, I will request you, and if you, by giving pause you will draw the diagram also. This diagram I have drawn by very simply, each and every points and each and every topics are given, and this is the elaborate, all the cellular organisms are there. So without a cell, a body cannot be exist. At least one cell must be there, or more cells are there. Each and every point I will explain you. First of all, you see the preview, what things today I am going to explain. The cell, how from a single cell a tissue is formed, then from the tissue organ, then organ system, organism. Next about the cell, then cell theory, then unicellular, multicellular organism. These are the unicellular and multicellular diagram, cell shape and size. Then animal cell, plant cell. Then about the cell membrane, cell wall, nucleus, flagella, cilia and flagella, mitochondria, vacuoles, ribosomes, endoplasmic reticulum, Golgi body, centrosome, lysosome, plastids, animal cell plant cell difference, cell division, mitosis and meiosis. Today also I have explained a little bit, although these are not included in your syllabus, then also I will try to explain a little bit about mitosis and meiosis. Then today I will explain the practical also, that is how plant cell, that is onion cell, has to be prepared in the laboratory. Then onion cell diagram, then the chick cell, how chick cell is prepared, chick cell structure. Up to this I will explain. So cell, from one cell, whenever a collection of certain cells of similar shape and structure, they are forming a particular function, they are called tissues. Some tissues together forming an organ, and some organs will form the organ system, and some organ system together forming the human being or an organism. Next you come to the cell. Cell is a structural and functional unit of life, as I have explained earlier. That means it is the unit. It means only one cell is forming one unit of the cell. Cell is a building block of life, means that anything less than a cell or anything less than a complete cell does not sustain life. Life cannot exist without at least a singular cell. Cell was first discovered by Robert Hooke in 1665. Next about the cell theory. This is very important. Several times question asked. So Sladen and Schwann, they have given three principles. Every living organism is made up of either one or multiple cells. Cell is the basic unit of structure and organization in organism. Cells always arise from the pre-existing cell, which means new cells arise from the division of old cells. That is, cell cannot be prepared artificially. Only cell can be formed from the pre-existing cell. That is, older cell, from older cell new cells will formed. Unicellular organisms, the organisms which are made up of only one cell, For example, amoeba, paramecium, euglena. Here you see the diagrams. You may pause the video and you draw the diagram. There is the amoeba, paramecium and euglena. You see they perform all the functions of their body with the help of only one cell. These are the multicellular organisms, a number of multicellular organisms I have shown here. It may be plant or it may be animal. So, here you see the diagram, that is bacteria, paramecium, amoeba, chlamydomonas. These are all unicellular organisms. Multicellular organisms I have shown: octopus, petromyzon. These are the shape of the cells. The shape of the cells may be oval, round, irregular, then elongated. These are the different types of cells with their shape. So, animal cell, this is an animal cell. About animal cell, I will take the help of the board, which diagram I have shown you earlier. From there, I will explain the animal cell. These are the cell, that means animal cell and plant cell difference. So, this is the cell membrane. Cell membrane is made up of phosphorus, that is the lipids and protein. About cell membrane, I am coming later on. First of all, you please see the board for the details explanation of the cell. Now you see the diagram, detailed diagram of cell as I have drawn myself, and it is the diagram of animal cell under electron microscope, although it is the diagrammatic representation. So, here you see this is the cell membrane. Cell membrane is bilayered. So, outer cell membrane and inner cell membrane, two layers are there, outer and inner cell membrane. Some pores are there, these pores are called the cell pore. Next part, that is the, here you see this is the cytoplasm. So, since this is cytoplasm, outer side is solution or water like, therefore it is called cytosol or ectoplasm. Cytosol or ectoplasm. Inner side it is just jelly like, as we are taking jelly with bread, just like we see this is jelly like, therefore it is called the cytogel. So, cytogel or endoplasm. Endoplasm and ectoplasm. Endoplasm, ectoplasm together forming the cytoplasm. Inside the nucleus there is a nucleoplasm. So, nucleoplasm and cytoplasm. Nucleoplasm and cytoplasm, that is all the liquid part of the cell, that is called the protoplasm. So, protoplasm is called the physical basis of life. Very interesting fact is that you try to remember, this protoplasm is called the physical basis of life. This protoplasm, if will heat, then it will coagulate, that is it will form solid. For example, you have seen an egg, you imagine an egg, inside the egg whole protoplasm, this one will, whenever will heat, this will solidified, and whenever will, that means shaking it, and then ultimately it will forming to solution like. And inside this cytoplasm there is, try to remember, there is an zigzag motion, that motion is called Brownian movement. This motion only we cannot give artificially, therefore we cannot make a cell. Next, here you see this is nucleus, nucleus made up of nuclear membrane, then these are the chromatin reticulum, and this is chromatin reticulum, this net like structure, this is called the chromatin reticulum. Some granules are there, these granules are called chromatin granules. Chromatin reticulum, chromatin granules, this is the nuclear membrane, and nucleoplasm. Another is the nucleolus, this is a large structure, hard structure, that nucleolus together forming the nucleus. So nucleus is the controlling center of the cell. One by one I will show in the slides the individual diagrams also. About the function of these things I will explain later on about the whenever I will show the individual slides. So this is called mitochondria. Mitochondria is the powerhouse of the cell. About the ultrastructure I will draw later on. This is the lysosome. This is ribosomes. Here you see ribosomes are attached with the endoplasmic reticulum. Don't confuse endoplasmic reticulum and chromatin reticulum. Chromatin reticulum and endoplasmic reticulum. Both of them are reticulum. Chromatin reticulum are present inside the nucleus. Endoplasmic reticulum is present inside in the cytoplasm. These two things several times you may be confused. And this chromatin reticulum will form the chromosome, and that chromosome will help for hereditary transmission from one generation to another generation. So you just see, this is the diagram. Next, lysosome. These are the ribosomes. Whenever ribosome is present, then it is rough. Then it is called rough endoplasmic reticulum. If ribosome is absent, the places where ribosomes are absent, these are called the smooth endoplasmic reticulum. So SER and RER, these are written in your book. So endoplasmic reticulum, rough endoplasmic reticulum and smooth endoplasmic reticulum. Next you see here, this is the centrosome. Centrosome is attached with the nucleus. That is the nuclear membrane and centrosome is attached with each other, and this centrosome I have drawn here by the enlarging, more enlarging. This is the nuclear membrane. This is nuclear membrane. With nuclear membrane this centrosome is attached. Inside the centrosome two centrioles are there, and these are the Golgi bodies. Golgi bodies is attached with the centrosome by thread-like structure. These are the protoplasmic thread. These protoplasmic threads are called the astral fibers. These astral fibers will form astral rays at the time of cell division. That is, the chromosomes will attach with the astral fibers, and ultimately they will divide from one cell to two cells. That is, one chromosome to two chromosomes, and ultimately, for example, in our body there are 46 chromosomes. These 46 chromosomes will divide into 46 and 46, that is 92 chromosomes. Again 92 chromosomes, they are going to two different poles, and ultimately they will form two cells. About these things I will explain in the slide. Next, structures. About the structure I have explained nicely. So each and every structure I will explain in the slides. So we try to remember about the structure of the cell. So now come to the cell membrane. As I have explained, cell membrane is made up of double layer. So cell membrane, this is the external layer, this is the internal layer. So bimolecular lipid layer, you can see these are the lipid layer, this thread-like structure, two thread-like structure, these are called the lipid layers, and these are the proteins. So these are the proteins, this is the intrinsic protein and these are the extrinsic protein. About these things we will study later on. So proteins and lipids together forming the membrane, cell membrane. So these are the peripheral proteins, you see, peripheral or extrinsic protein, and this is called integral protein or intrinsic proteins. These are the lipids. So bilayer lipid layer and monomolecular protein layer. Next come about the cell wall. So cell wall is dead. So structural layer next to the cell membrane is called the cell wall. Cell wall is present only in the plant cell, but in animal cell there is no cell wall. Animal cell directly coming to the cell membrane. So next you come to the nucleus. As I have explained earlier, nucleus, this is the diagrammatic representation. Nucleus, there are nuclear pods. We have seen these are the nuclear pods, and through where the chromatin reticulums is there. You see the diagram which I have drawn in the board. This is the nucleolus. These are the chromosomes or chromatin reticulums. This is the nuclear membrane and nuclear pods. Chromatin reticulum, This chromatin reticulum will form chromosomes at the time of cell division. Next you see, these are the cilia and flagella. These are present in your syllabus. Cilia is present in the paramecium and flagella in euglena. So flagella, you will see the size of the flagella, that is this whip-like structure. This structure is larger than their body. When their size is more larger than their body, this is called flagella, and these are small outgrowths or hair-like outgrowths. These are very small and they are present. Whenever they are small, then they are called cilia. Cilia and flagella, both of them are responsible for movement of the organism from one place to another place. Next, come to the mitochondria. So mitochondria is called the powerhouse of the cell. So inside the mitochondria, here you see this is also double layered, outer membrane and inner membrane. This is the outer membrane and this is the inner membrane. This is called the inner membrane. And here some mitochondrial ribosomes are there. These are called cristae or cisternae. And inside the mitochondria, ATP will form, adenosine triphosphate. Here you see this is the matrix, the liquid substance which are present inside the mitochondria is called matrix. Inside the mitochondria there is also mitochondrial DNA. About this structure you will learn later on. Now you just try to remember mitochondria is called the powerhouse of the cell. It is forming the ATP. ATP is the energy currency. That is, ATP will store energy. Whenever we need energy, then ATP will provide energy and forms ADP. And whenever there will be excess energy, that excess energy will form ATP from the ADP. So try to remember about the mitochondria. Mitochondria is called the powerhouse of the cell. This is the vacuoles. Vacuoles are mainly present in the plant cells. Larger, and animal cells also vacuoles are present, but animal cell vacuoles are very smaller and the plant cell vacuoles are very larger. This is the ribosome. So here you see ribosome is called the protein synthesizing factory of the cell. You may write in your copy or you may remember, that is the ribosomes are called the protein synthesizing factory of the cell. So this is ribosome. So in ribosomes, what things we are seeing? Ribosomes in forming these polypeptide chains or proteins. Ribosomes are present in endoplasmic reticulum. Here you see this is the nucleus and here is the endoplasmic reticulum. This endoplasmic reticulum, whenever ribosome is present, then it is called the rough endoplasmic reticulum. Whenever ribosome is absent, then it is called the smooth endoplasmic reticulum or SER. Here you see this is endoplasmic reticulum and how ribosomes are attached with the endoplasmic reticulum. These are the ribosomes, these are attached to the endoplasmic reticulum. Here you see nucleus and then from nucleus ribosomes are coming. The endoplasmic reticulum is coming and inside in the endoplasmic reticulum these ribosomes are attached. This is Golgi apparatus. So Golgi apparatus is helped in secretion and transportation of the substances in the cell. Another thing, as I have explained, endoplasmic reticulum helps to enter and exit of the substances. Here you see this enter and exit of the substances inside the nucleus or from the nucleus to cytoplasm. You see the diagram, then you can understand. Next, this is the centrosome. So centrosome, as I have explained, centrosome and centrioles, these centrioles are here you just stacks, just like these stacks of thread like structures, and centrosome is helped at the time of cell division. This from the centrosome. Here you see in this diagram from the centrosome. This is the chromosomes. So chromosomes and centrosome, these are attached by the astral fibers. As I have explained in the diagram, astral fibers help to attach the chromosome with the centrosome. So these things are helped in the at the time of cell division. This is called lysosome. Lysosome is called the suicidal bag of the cell. Lysosome is actually an organelle which secretes enzymes, which is called the lysoenzyme. Try to remember lysoenzyme. So lysoenzymes helps to digest, cellular digest inside the cell, whatever the substances are there, that things will absorb by the or that is the digested by the lysoenzyme. So whenever there is any wound or cut in the cell, that means in the tissue, then the cell, inside the cell, the lysosome wall, this wall of the lysosome, here you see this is the walls of the lysosomes, these are the walls will burst. So if the walls will burst, then the lysoenzyme, whole the lysoenzyme, these lysoenzymes will come out in the cell and ultimately dissolve the nuclear membrane. Whenever these lysoenzymes will come out in the cell, that is in the cytoplasm and protoplasm, then what happens? This nuclear membrane will dissolve and the whole nuclear material will come in the cell, and then the cell will die. If the cell will die, then what happens? It will help the nearby cells. Therefore, lysosome is called the suicidal bag of the cell. By suiciding, by the sacrificing of its own life, then what happens? It will help the other cells, nearby cells, and then the bacteria or other microorganisms cannot enter inside the cell, and the other tissue will saved. Therefore, lysosome is called the suicidal bag of the cell. Next, plastid. Plastid is present only in the plant cell. In the animal cell, plastids are not present. So plastids are mainly of three types: chloroplast, leucoplast, chromoplast. Chloroplast is the green color plastid, chromoplast is the yellow or colored plastid, and leucoplast is the colorless plastid. These are present only in the plants. They are help in the photosynthesis. Inside the plastid, you see there are the stroma and grana. Two things are present. Plastids, this is the structure of chloroplast. Plastid also bilayered or double layered. These are the difference between the animal cell and plant cell. Animal cell and plant cell, you see in the animal cell there is no cell membrane, whereas that is there is in animal cell there is cell membrane, but there is no cell wall. But in the plant cell there is cell wall, and in addition to cell wall there is cell membrane. Animal cell, that is dictyosome, is present in the plant cell. In the animal cell there is the centrosomes, cell wall, chloroplast and vacuoles. These three things mainly present in the plant cell, but in the animal cell these are absent. And the vacuoles are very large. Here you see this is so large vacuole, and this is smaller one is the nucleus. Here this is the nucleus. So vacuoles are absent, or sometimes may present vacuoles. Small vacuoles are present in the animal cell. Cell division, that is from one cell, these are present in your book. When from one cell to two cells are formed, this is called the cell division. Cell division is mainly two types: mitosis and meiosis. Mitosis and meiosis, these are not included in your syllabus, but I am explaining a little bit. In mitosis means the somatic body cell. Somatic means body cell divide. Our body cells divide from one cell to two cells. Only the difference you try to remember, that is here you can see the two chromosomes are there. In the daughter cells, these are the daughter cells. In the daughter cell you are getting same two cells. From two cell, that means from the one new, there is two chromosomes, there is two chromosomes in one cell, there are two chromosomes. Just this hypothetically we have taken. Since it is two chromosomes, in the daughter cell also there are 2 chromosome. Daughter cell 2 cells are there. In both the cells there are 2 chromosome. That is, chromosome number will same in the daughter cell as their parent cell in case of mitosis. Therefore, from this daughter cell, this will again form like larger cell, and this will form into 2 cells, and the chromosome number remains constant in case of mitosis. Whereas in meiosis, from one cell 4 cells are formed, but the chromosome number reduced to half. Therefore, it is called reduction division. Here you see there are four chromosomes. From the 4 chromosomes, each and every cells are forming 2 chromosomes. Therefore, the chromosome number divides into half. So, I am explaining, just you try to imagine our body cell possess 46 chromosomes or 23 chromosomes. So, body cell in earlier, that means what things we have seen, if it is here it is given 1 pair, but our case present 23 pairs of 46 chromosomes. From this 46 chromosomes, the daughter cell also 46, therefore same characteristics. Then this cell again, this one will be divide and forming 46. But in case of meiosis, from 46 chromosomes, the daughter cells are forming 23 chromosomes. So these things occurring only in our reproductive cells, that is, in case of male forming sperm, and in case of female forming the egg. So sperm and egg, they are present only 23 chromosomes, but our body cells possess 46 chromosomes. So whenever one sperm and one egg, that is 23 and 23, they will fertilize, that is, they will unite and they will fertilize, and then they will form 23 plus 23 equal to 46 numbers of chromosomes, and just like body cells, somatic cells. So our, that is, from mother's body coming 23 chromosomes and father's body coming 23 chromosomes, and ultimately our body forms 46 chromosomes. Next, this is the practical, whatever you have done in your practical classes, how the slide is prepared. I am showing this is additional. You will take an onion, and these are onion pieces, and you will just break it, and then you will peel it. By peeling, you will give here, and give in the slide you will give some saffronin, and you will giving by some water, you will wash it, and then just you will cover it with a cover slip, and then you will see in the microscope. Whenever you will see in the microscope, then you will see like this. These are the cells, the cell wall and cell membrane. It is cell wall, nucleus, it can be seen clearly. Probably you have done in your practical classes, or in class 9 you will do the practical class. This is the cheek cell. How you can prepare cheek cell? You will just take a plastic stick or plastic scale, and from your cheek, inside the buccal cavity, from your cheek you will take some amounts of scratches, and you are putting in the slide, and by giving some stain and in the cover it by a cover slip, and then whenever you will see, then you will see like these cells. These are the animal cells. So this human cheek cell, it is animal cell, characteristic animal cell, and that was the plant cell. This is the characteristic plant cell. Therefore cell wall is there, but here there is no cell wall, only the cell membrane. So today up to this, and now you come to the board. So today probably you have understood everything about the cell. You will draw this diagram nicely, all the things you have seen properly. So today what things we have studied about the cell tissue, organ system, organism, then about the characteristics of cell theory, unicellular organism, multicellular organism, then about the drawings of unicellular and multicellular organisms. You will pause the video and you will draw this diagram, then shape and size of the cell, animal cell, plant cell, cell membrane, cell wall, nucleus, then cilia and flagella, mitochondria, vacuoles, ribosomes, endoplasmic reticulum, Golgi bodies, centrosome, lysosome, then plastids, then the difference between animal cell and plant cell, cell division, mitosis, meiosis, and at last the processes of preparation of onion cell and human chick cell. So today up to this you have studied properly. You please see several times the video for understanding, and probably most of you can answer any question from here. If you have any doubt, then you please call me or you may contact me and you may write the comment. If you like my video, then you please share and subscribe. Thank you.